It's a typical Monday morning at Waikiki Beach. Towels and umbrella chairs crowd the sand. People of all ages lie out, letting the rays of sunshine penetrate their skin. Surfers wait at the brake line for a ride to shore. Sailboats clear a path through bobbing heads and plastic rafts. On any given day, there are about 220,000 vacationers spending millions of dollars in the state of Hawaii. And most of these people come for this scene at Waikiki Beach and to snorkel or go on a turtle watching tour, which means coral reefs play a big role in this piece of the Hawaii economy. So with warming seas threatening coral reefs worldwide, scientists are now looking for ways to save this valuable resource. From Currently Ocean News, this is Wavelengths. I'm your host, Betsy Brown. A quick note before we go much further, if you don't know much about coral bleaching, I recommend you listen to the episode White as Snow, Coral Bleaching in the Pacific before continuing with this one. That presents the problem. This episode looks at a potential solution.
This is ambient, so I have actually some of my Montifera capitata.
Beth Lenz is holding the hope of Hawaii's coral reefs in one hand.
But like I said, they grow so slowly.
It's a tray with several circular surfaces. She points to a tiny bump on one of the circles. It's barely bigger than a grain of sand.
One right there.
Oh, wow. Beth is a PhD candidate at the University of Hawaii. We're standing in a small trailer-like building. The wall is lined with white tanks. They're open at the top and filled with water. All of them are connected to a computer that controls and monitors their temperatures. The tray with the tiny bump came from one of those tanks. What are they?
These ones are from June.
Oh, wow.
So let's see if we can. So if you compare.
It's Montifera capitata, commonly known as rice coral. And Beth hopes it'll grow into a super coral of sorts, able to survive higher water temperatures than normal.
So for all of my experiments, I've been using this specific parental colonies based on their bleaching history, whether or not they had bleached, and then taking their eggs and sperm. So if you have an egg donor or a sperm donor based on their bleaching history, can you raise more successful larvae based on their parents?
Beth is one of the many scientists working under Dr. Ruth Gates at the Gates Coral Lab. It's located on Coconut Island, a tiny speck of land in Kaneohe Bay, where the Hawaii Institute of Marine Biology has all of its research facilities, and where Dr. Gates and her students are trying to get ahead of climate change. Kira Hughes is the research project manager there.
So there's three main parts of our project. One is selective breeding, which is what Beth does. And then another is inducing acclimatization, so that's letic conditioning. What doesn't kill you makes you stronger. And then the third is modifying symbioses. So corals have these little algae symbionts that live within their cells. And so if you have hardier algae that lives within the cells, then maybe those corals will not be as likely to expel them, which is the reason that they become white in the first place.
What they're doing is called human-assisted evolution. It's a little controversial in the science world.
It doesn't mean doing GMOs or anything like nothing is genetically modified. It's just actually speeding up a process because the corals are not keeping up with what's changing with the environment. They can't keep up with the fast rate that it's changing. You know, there are some people that say, well, you might be minimizing diversity by only, for example, what Beth does, only breeding specific genotypes to be able to get those hardier corals. But we'd rather do that than not have any corals at all.
If you look at the long list of Dr. Gates's publications, you see she was interested in coral bleaching long before it was recognized as a major problem. Then in 2014, things got urgent. That's when the National Oceanic and Atmospheric Administration says the third ever multi-year global bleaching event began, and it continued through 2017. In Hawaii, bleaching was recorded in 2014 and 2015, some of it in Kaneohe Bay, right outside the Gates Coral Lab, and they noticed something peculiar there. Some of the coral bleached, while colonies of the same species seemed unfazed by the changing temperature.
Why would one coral colony bleach when it was right next to one that didn't? So why is one coral colony more resilient than the other? So that's kind of the big question that we're investigating.
The global bleaching event got the world's attention, making it easier for the lab to find funding for experiments that had already been planned out. And by using the coral in Kaneohe Bay, they were able to accelerate the project.
Because there was a bleaching event that allowed us to already identify the bleached, non-bleached colonies instead of doing a manipulative experiment in the lab and doing a stress test.
And then we've been working with those more resilient corals to try to get these super corals that we call them, where we already know that they're tough, then if we breed those kind of super athletes, then will you get offspring that's even stronger? So what was really interesting is doing a stress test with those genotypes that we worked with that we know are already more resilient, and we could not get them to bleach. There was only one genotype out of 20 that would actually bleach when we were just pumping the temperature up. So it's really reassuring that we are already working with more resilient corals, and then we're hopefully going to be able to grow ones that can be even more resilient.
All of this research takes time, and for Beth's part of the project, that means following the coral's biological clock.
So depending on the species, depending on where you're located, corals will only have a certain time on the entire year that they release their egg, sperm, or their larvae, depending on the species. And so during a coral spawning season, you only have maybe 1 week within 1 month between 2 months to actually get your material and do your studies. So that is a huge limitation in itself.
I wasn't at the lab during the magical window of time to see the coral spawn. That happens in the summer. But here's how it works with rice coral. The coral releases little bundles of eggs and sperm. They look like tiny hot air balloons floating towards the surface.
And then that bundle dissolves in the water, and then the sperm swim about and the eggs float to the top to get fertilized.
Beth showed me a video of the spawning to help explain how they do the breeding.
So we collect those bundles, let the eggs and sperm break apart or separate, and the eggs will float to the top. The sperm will sink a little bit, but we can pull the eggs out and then do our intentional crosses.
Okay.
Yeah.
Since they hadn't done any intentional breeding in the lab before, Beth says they were learning these processes as they went.
And so we've been able to push through in the last 2 years now. We were able to successfully get them fertilized, raise the larvae, get the larvae to actually settle, because there's all these different life stages for a coral. It's a really slow process, and it's really sensitive, especially since the coral that I'm working with, Montipora capitata, the rice coral, actually has an acid inside of the egg too, so you have to be really gentle.
Montipora capitata also grows very slowly, but another coral the lab is working with grows much faster. Back in that trailer-like building, she shows me coral that was bred at the same time as the tiny coral I saw.
If you compare, let's say, these that were from over the summer versus, one of my small ones, you can already see quite the difference.
It's those little dots at the bottom.
It's those little dots. And this is Passalopora acuta versus Montipora capitata. And so we're finding huge differences in growth.
It's like seeing a pebble next to a grain of sand. Ariana Huffmeyer works with that faster-growing species.
The lace coral, Passalopora acuta.
She's studying how the environment affects them from that very young juvenile stage.
But it's a really important, really critical life stage because corals need to pass through that juvenile life stage to enter as a reproductive adult.
Scientists don't have a handle on when the coral she's working with actually enters the reproductive stage, so there's still a long ways from seeing if the resilience can be passed from one generation to the next.
And that's something that over time, as the juveniles that I'm working with grow out, we'll actually be able to see potentially when they start releasing larvae and be reproductive. But right now, we really don't have a good handle on exactly how long it is.
So for now, Ariana is working to answer questions about an individual coral's ability to adapt. She explained an experiment she did where the juvenile coral was placed in various conditions.
So I had a temperature that fairly represented what we see in the natural environment during the summer, and one that was slightly cooler than that. And then individuals within those two treatment groups were either given food or not in the form of just natural plankton that you find on the reef.
And the coral that was exposed to the better conditions at an early age was less affected by heat stress.
Corals that were provided an extra source of food in the form of plankton grew faster. They had thicker tissues, more symbionts. We're still analyzing some of that data, but that's kind of what it's showing right now. And the corals that were in a slightly cooler condition and provided with food did better in thermal stress than any other group. So there's some evidence that just from that experiment that over the first month of their life, that if they're exposed to different conditions, that influences their stress response.
The experiment, she says, brings up important questions about water temperatures throughout the year.
We know that summers are getting warmer, but winters may also be getting warmer, and so we're losing some of that cool period that corals are exposed to on an annual basis. So we can start to ask questions about how that cool period really plays a role in their thermal resilience and that bleaching response. It may be that cool period is more important than we originally thought.
That's information that might apply to coral in other parts of the world. And that is exactly the kind of impact the Gates Lab hopes their research will have. So that all the economies that rely on coral for tourism, food, and coastal protection will have it in the future, no matter how hot the water temperatures get. Wavelengths is a production of Currently Ocean News, produced and hosted by me, Betsy Brown. Theme music is by Sean Kish. If you'd like to learn more about the Gates Coral Lab and the research that they're doing, you can go online to wavelengthsoceanpodcast.com. And if you haven't done so yet, please rate Wavelengths on iTunes. It will help others find the podcast.